I didn't realise this today, but it turns out I'm from the Jeff Goldblum School of Statistics. So really what I want to address is, clearly we can do this, but I really want us to stop and pause for breath to think whether we should. I confess I find the whole area fascinating. I've really enjoyed trying. I can't say I've been particularly successful trying to keep up with the amount of literature there is in this area. And I've had many hours of fun trying to come up with the optimal testing strategy to work out how I can best spread my bets so that I have the best chance and I can guide the team to have the best chance of being successful. And indeed, at AstraZeneca, we've decided on the basis of conversations primarily with FDA to do this. But nevertheless, I've always had this nagging doubt as to whether we really need to go as far as we're doing. And I've ended up looking at really trying to question the assumptions behind why we're doing this and whether it is indeed the optimal approach. It is clearly complicated. It also can be counterintuitive. Now, just because something's complicated, it doesn't mean we shouldn't do it. But I think it does mean we have to ask you whether it's worth the complication. And that's what I want to ask today. But if we conclude it is worth the complication, then we have to recognise that means there is a lot more work on our behalf needed to educate and justify why we're doing it to others. So, as I say, really what I want to do is just to pause for thought and question, and I hope provoke some debate as to whether we should do this, not so much how we do that. I clearly will defer to others, especially many people in this room, as to how we should best do it. So, disclaimers get everywhere. Next slide. This is really, I find the terminology here rather unfortunate. And I reflected on this as I was doing my slides when I was getting my car serviced. And as the cost and the garage kept calling me for other items, we're going to add more to the car service, they asked me, so you need some more brake fluid. Would you like some more? I'm thinking, well, I don't know much about brake fluid, but it sounds like I should have strong control of my brakes. But I don't know anything about brake fluid. And so when I'm asked, would I like weak or strong control, sir? It's very difficult to say I would like weak control. So in my presentation today, I'm actually going to offer that weak control with some modifications in this context, unlike probably is the case with brake fluid, but I'm never quite sure about garages, that in fact is the case. But it does make, I acknowledge, arguing the case a hell of a lot harder when my proposal is to use weak control. So these are the questions I want to address. Could, in fact, the level of complication, if we're not careful, become self-defeating? So it's exactly the thing we're trying to control for actually doesn't happen because people disregard what's done and apply their own clinical judgment anyway because the whole thing doesn't seem to make sense to them. And therefore, we should question whether the extra complexity is worth it. But then it made me think, what are we actually trying to do with these approaches? First of all, we have a decision to make as to whether we should license the drug. And by and large, that's going to be based on the primary endpoints. And absolutely, we should have full control amongst those primary endpoints. Then, what role do the secondary endpoints play? In my mind, if we've established amongst the primary endpoint that my drug works and the benefit offsets the risk, which I'm looking at independently and don't apply any multiplicity adjustments to, then the secondary endpoints, their role in the prescribing information and the licensing is to describe to the prescriber what I think the nature of those benefits or whether there may be some additional benefits to those drugs. So really this slide repeats what I've just mentioned, but just to say, deciding talks about, in my mind, what we do with the primary endpoints, and the secondary endpoints are all about, describing what we do and describing the nature of that benefit. I'm not going to belabor the point on this slide. Everybody's very familiar with this, but just to re-emphasize again, I certainly do think we should be absolutely rigorous amongst our primary endpoints and control type 1 error. As an aside, we've not mentioned this today, I know it's not the topic, but we should be even more concerned that aspects of the design and how we conduct the trial and include missing data in that are absolutely flawless or else everything we're talking about here with multiplicity and controlling type 1 error will be irrelevant because the significance levels won't have the level of control we think they have. So some examples of weak control. Again, everybody's very familiar with that. But if I have weak control, it still guarantees there is, in the decision whether to license the drug, I have a fully controlled 2.5%, 1% alpha, for example, that I fully controlled the decision as to whether falsely to license the drug. So what's strong control? The definition at the top there is the probability of rejecting any. I want a more true null hypothesis is at most 2.5%, irrespective of how many and which hypotheses are actually true or false. So we need to consider every single possible permutation amongst the tests that I'm testing of permutations of those of which are true and false, and I have to control type 1. I have to have a strategy that controls type 1 error in all those situations. So it involves testing the treatment effect amongst those hypotheses that are true. We will never actually know for certain which those are, even at the end of the trial. And the other part of this has always felt slightly uncomfortable to me is the fact that if, for example, we are to assume amongst the primary endpoints the null hypothesis is true, then actually I don't need to do anything with my secondary endpoints because the probability of falsely rejecting a true null hypothesis will still be 2.5% if I am prepared to say a secondary endpoint can never rescue a failed trial amongst the primary endpoints. So the only time, the reason we need to adjust secondary endpoints, the only time we need to do it is if we're prepared to believe, before we've done the trial, that the hypothesis, that the null hypothesis is not true on at least one of the primary endpoints. And that's the source of why we need to do this, because if that's not the case, then just making a decision amongst the primary endpoints is sufficient. So here's some examples. I'm gradually going to build these up. Anybody, and it's been mentioned already, anybody who's worked in this knows there's a real tension in terms of where we order the endpoints between choosing those that we would really like a claim on that are very difficult, such as overall mortality, versus those that we think are easiest to get. But if I go for the ones that are easiest to get, what if I lose out on the thing I really want and in some sense I get lucky? So you could go either way. Clearly you can make this more sophisticated, and this tends to be, looks very similar to how we've addressed this within oncology if PFS is a primary endpoint. But it does get more complicated. The next slide has really been covered already. So this is a case, this is a real example. We had one control group, 2 experimental arms. One was the drug given in monotherapy and the other drug was the drug given in combination, addressing 2 quite separate questions. But I was to learn as I came to this that this was the first example of many of the testing approaches I'd previously assumed were okay, such as Holm or Hochberg procedures, actually break down in this context. So for example, if I found out my combination, say my primary endpoint was survival, I'd adjusted my significance level, but my combination was still significant. If I then pass all my alpha, recycle all that alpha to the monotherapy comparison, as I may do in a Holm procedure or step-up procedure, if that monotherapy fails, then according to this framework, I can't make any claims that any of the secondary endpoints were significant. So for example, I would have to say to a physician, you've got a survival advantage, PFS, the p-value is vanishingly small, the treatment effect's clinically important, but you can't make a claim on that. Now clearly there's a solution, you pass some of your alpha straight to the secondary endpoint, but it does mean, as has been mentioned already, I can't use procedures such as Hochberg or Holm procedures amongst my primary endpoints in the strong control framework. It also means, incidentally, other approaches that sound, on the face of it, very reasonable to say that if both of my endpoints were significant at 2.5%, could I then test separately both my combination and monotherapy hypothesis, both at 2%, 2.5%, because I've now proved on the primary endpoints they both work. That doesn't work either. That doesn't strongly control type 1 error. So an example now, This just as an aside, just remind me, I'm going to present it in the way that Carl Frederick Berman uses this, but I'm told that it's exactly the same as the Brett's approach. The approaches are the same. But it reminds me of a game called Rummikub, which my children of 10 and 12 very much enjoy. It still amazes me the extent to which their father is ruthless in wanting to beat them in this particular game. So this is an example. I won't go through it, but this is a real example of a trial we were going to do, but we didn't in the end for various reasons. It actually was a little bit more complicated than this. So this is how we decided to kind of split the difference. We would, for some comparisons, we would go straight for the far right-hand side, straight from the PFS to overall survival. But sometimes we'd then split alpha, recycle alpha from the combination to the monotherapy, which is the comparison we most wanted to win on because it would have better toxicity. So I'm going to skip through these slides. If you suffer from motion sickness, I suggest you look briefly away. These will be on the slides, but this shows you really how it works, but I won't belabor the point. Okay, I wish I hadn't put so many builds on now. Right, now another example here is when we start having more experimental dose arms. Clearly it looks very complicated. This has been optimized to increase our chances of success amongst the various endpoints and the different experimental doses. It doesn't have to look as complicated. We could summarize it in a diagram such as that, but in reality you have to work through the left-hand side to decide whether something's significant or not. So now on to group sequential designs. I spend most of my time working in oncology, and this is where things get even more complicated, although I have to say some authors, and I think one of the authors is in this room, has come up with some very elegant solutions. So there was an initial paper by James Hung which asked the question, well, if I'm going to do 2 analyses of survival and 2 analyses of progression-free survival, can I test PFS at 2.5% one-sided at either the interim or whenever survival is significant? So if survival was significant at the interim, can I test PFS at full 2.5%? Whereas if it was significant at the final, could I test it at 2.5% in that case? Now it turns out, and it was highlighted, no you can't, not to strongly control type 1 error. And a very elegant solution was provided by Glim. However, what I want to do here is then to really look under the bonnet of that and say, well, how much difference is that making? Because it then now brings in a new component. I need to consider how strongly correlated my PFS and overall survival endpoints are, and that governs the extent to which type 1 error may be increased. So on this plot, let me just explain it to you, is along the x-axis is the true hazard ratio for the primary endpoint. Along the y-axis is the correlation between the 2 endpoints, which of course I don't know. And then on the z-axis going upwards is the one-sided type 1 error. Now, as you can see, in most scenarios, there is no material effect, if any, on the type 1 error. It's only on a particular point on the graph at a particular true hazard ratio and extremely high correlations, could there be any material increase in type 1 error? And even in then, it's probably only gone up to about 4% from 2.5%. So we impose this very, very complicated procedure when there's very little difference. And when I went back and looked at some cases in cancer where I thought the correlation would be its highest, when there was a very, very small time between progression of disease and death, sadly. But even in those cases, the correlation is not much more than about 0.5, and that's nowhere near the area where there's any appreciable increase in type 1 error. And in fact, in that area, the maximal alpha is 2.8%. So this is certainly an area when you try and solve this problem, it gets very complicated. There is a solution. If you then also apply a spending function to the secondary endpoints, you look at the same graph. I always control type 1 error. But I can assure you, if you've got multiple dose arms, or multiple experimental arms, and you're analysing the data more than once, then you bring in a whole level of complexity. And then there are practical questions to answer, because often we may analyse PFS and response rate once, but we analyse survival once, we analyse it later. So if I'm only going to collect the data on PFS once and response rate once, do I need to adjust them even though I'm testing them once? I don't think so, actually, but when we've discussed that with regulators, they've not accepted that. Or can I test them twice, even though I've got the same data set? Do I need to adjust then? So those are some of the practical questions we need to consider. And it does lead you to think, for example, on the testing procedures, if I'm not going to use full 5% on survival, which might be a secondary endpoint, do I compensate the trial design there to either increase patient numbers or get some extra events and wait longer? So that's indulgence over. So I've offered a few questions. A question, well, what do you suggest instead? What's your alternative? A very reasonable question. I really said this before, but I think that the key thing is that the bottom 2 points there in purple is it's really driven by what are we trying to do. We've got a decision to make whether to license, and having done that, we then need to describe the nature of the benefit and risk. Sorry, I forgot my next slide. Here's a real example. So this is very important. This is the Brilique SPC. This is taken directly from the SPC. And I've pulled out all-cause mortality. The p-value is very small. And appropriately, the SPC indicates, according to the predefined hierarchical testing, that's not significant. So that's what's in the SPC. Clearly, the p-value is there as well. And interestingly, when you look at the EPAR, here are some direct quotes, and I think this is an entirely appropriate interpretation of the data. All-cause mortality was also significantly reduced. Black and white statement, even though according to the testing procedure it wasn't. The most important secondary endpoints are supportive for the primary endpoint, including all-cause mortality. So is all-cause mortality a special case we need to talk about? Although in this situation we had a testing procedure, but of course a judgment, and I think common sense, was made in terms of interpreting the data we'd got, despite the testing strategy we'd produce. So what would I do? Absolutely, I think we should rigorously control type 1 error amongst our primary endpoints. But then, amongst the secondary endpoints, agree what are the important additional questions that I'm going to answer. And we shouldn't go and then and answer those 10 different ways and choose the best. We should choose, well, which endpoint or set of endpoints best addressed those questions. But having done that, I'm not convinced there is then a need to further adjust the significance levels of those. And I have to say it was very heavily influenced by a paper I read in my formative years, for which I apologise, I don't have the reference, but it was by Cook and Fairwell. They published in the JRSS Series A publication, which suggests a model very similar to this. So primary endpoint, did the patient live longer? I may then have some other questions. Did I slow down the progression of the disease? Did the patient experience more tumour shrinkage? Did they feel better? I certainly think we should reduce that control alpha amongst that questions, because often a number of different ways of answering that. And just a final example, which I came across too late to get in the final cut of slides, was a drug called, making sure I get this right, Actemra, which was in the RA area, which is another good example. There was a public advisory committee. The company quite appropriately indicated which endpoints were significant according to the testing procedure. There are a series of 3 related endpoints: ACR 20, ACR 50, and ACR 70. ACR 20 appeared highest in the hierarchy. ACR 50 and 70 were further down and were not significant according to the testing procedure. But what's interesting, that despite that, in the FDA labeling, the ACR 50 and ACR 70, the p-values are all in there, the confidence interval, they're all in a label, presumably claims can be made, and there is no mention whatsoever of the fact that that was not significant according to the testing procedure, which suggests to me that the behaviour of the reviewer, again I think quite appropriately, was in more of this mindset. I've seen an effect on ACR 20, therefore it seems reasonable to me to describe the nature of the benefit on 2 very closely related endpoints, put those in the labelling, and interestingly didn't feel the need to indicate that formally that they were not statistically significant. So just in final, whilst the whole area is extremely fascinating, by that I don't want to demean what people have done, but I do think, just to come back to what Rob said this morning, is just because we can, it doesn't mean that we should. And I think we should consider a more considered approach to the role that multiplicity takes in the licensing in medicines and how we describe the nature of those benefits. So my hope was that this would prompt some discussion. I hope it does. And I'll be interested to see the extent to which I feel more comfortable with some of the proposed approaches after the discussion. So thank you very much.